Hey, Professor, could you tell me about the cell cycle?
Sure. The cell cycle consists of interphase and the mitotic phase. Interphase is the first and longest part, with 3 subphases. In G1, the cell grows. In S, it duplicates the chromosomes. And in G2, it makes some final preparation for cell division. The mitotic phase consists of mitosis and cytokinesis.
What if a cell didn't divide?
Excellent question. If a cell didn't go through the mitotic phase, asexual reproduction, growth and development, and tissue renewal or repair all wouldn't be possible. This is exactly why mitosis is so important.
So without mitosis, our injuries wouldn't heal and a child wouldn't grow into an adult?
Exactly. You're getting the hang of this.
Wait, so the nucleus just divides in half, then the cytoplasm splits and voila?
Not quite, if only it were that simple. Actually, there are multiple phases of mitosis. In prophase, the chromatin begins to condense into chromosomes, the nuclear membrane is degraded, and the nucleoli disappear. Also, by the end of prophase, the mitotic spindle begins to appear, the chromosomes begin their journey to the center of the cell, and the centrioles are positioned at opposite poles. In metaphase, the mitotic spindle forms, and the chromosomes are positioned on the metaphase plate. The chromosomes are each made up of 2 sister chromatids. In anaphase, the centromeres of each chromosome separate as the microtubules attached to the centromere shorten in length and cause the chromosomes to move to opposite poles. As soon as the centromeres separate, each chromatid has its own centromere and is now
a chromosome.
A chromosome, right. Lastly, in telophase, 2 daughter nuclei begin to form in the cell. By the end of telophase, the nuclei are ready for interphase of the next cell cycle. In animal cells, a ring of membrane, or cleavage furrow, pinches the cell in 2, and in plant cells, a cell plate divides the daughter cells. Okay, now it's your turn to answer questions.
Okay, I think I'm ready.
If a cell starts with 46 chromosomes, like a somatic human cell, how many chromosomes will each of the daughter cells have?
23. Oh, no, wait, 46. 46, because it contains 2 sets of chromosomes, 1 from each parent.
What is that called? Diploid or haploid?
Diploid.
Right. And is the cell genetically different from the parent?
Nope. Each daughter cell is genetically identical to the parent.
Wow, you're on a roll. Now, what about sex cells? They fuse together to create an embryo, so they must be haploid. How do they come about?
You got me there. How?
Meiosis.
How does meiosis work?
Meiosis is broken down into 2 parts, meiosis I and meiosis II. Meiosis I separates homologous chromosomes, while meiosis II separates sister chromatids. Homologous chromosomes are 2 chromosomes that make up a pair. In prophase I, chromosomes begin to condense, and homologous chromosomes loosely pair along their lengths. They become connected to each other in a state called synapsis. Also, crossing over occurs. This is when non-sister chromatids exchange corresponding segments of DNA. The points where crossing over has occurred are called chiasmata. In metaphase I, homologous chromosome pairs are arranged at the metaphase plate in a random assortment. In anaphase I, the homologous pairs separate by moving toward opposite poles. When telophase I is beginning, each half of the cell has a complete haploid set of duplicated chromosomes. Cytokinesis usually occurs at the same time, and 2 haploid daughter cells are formed.
And each haploid daughter cell is a complete set of duplicated chromosomes?
Exactly. Meiosis II accounts for this by separating the sister chromatids. This is how meiosis I and II are different from each other. The stages of meiosis II are very similar to the stages of mitosis.
So at the end of anaphase II, each sister chromatid is now a chromosome?
Correct. So how many daughter cells are produced as the result of one meiotic division?
Well, the 2 haploid cells from meiosis I each split into 2 haploid cells during meiosis II. So that would make 4 haploid cells?
You got it. So if a parent cell has 46 chromosomes,
if a parent cell has 46 chromosomes, each of the 4 haploid daughter cells would have 23 chromosomes, each genetically different from the parent cell. This is important because it introduces genetic variability in offspring. Otherwise, natural selection wouldn't occur, and offspring would not be able to adapt to a changing environment.
Wow, who's the teacher here, you or me? Very good. So can you summarize the difference between mitosis and meiosis?
Well, in mitosis, there are 2 daughter cells produced, each diploid and each genetically identical to the parent cell. This is important for growth, repair, and asexual reproduction. In meiosis, genetic variability is introduced. There are 4 daughter cells produced, each haploid with half the number of chromosomes as the parent cell, and each genetically different from the parent cell. Genetic variation occurs because of the crossing over in prophase I and the random assortment of the homologous pairs along the metaphase plate in metaphase I. Meiosis is important for producing gametes that can fuse to become new organisms and because it allows for genetic variation among these new organisms.
Fantastic!
Thanks for teaching me. Now I know all about the cell cycle.